So what is AIDS? AIDS stands for Acquired Immunodeficiency Syndrome. So AIDS clinically is a group of symptoms and diseases associated with a decrease in CD4 cells and the presence of the HIV virus. We'll talk about what that means in a little bit of detail here and now. So what are some of these symptoms? We'll talk about them as well, but things like shingles, so that's a reoccurrence of the chickenpox virus, various types of herpes, rare cancers, rare fungal infections, all sorts of weird diseases of the nervous, respiratory, and digestive systems. So generally, HIV and AIDS are linked in that HIV, if it is not treated, will usually progress to AIDS. So we'll talk about a little bit more about what the clinical definition of AIDS is. So what type of diseases are associated with AIDS when your immune system starts to get crippled? So disseminated herpes or shingles. So what does this mean, disseminated? Disseminated means it's spreading all over the body. So if you think about herpes, we've got herpes simplex 1 and herpes simplex 2. Generally, they are located orally or in the genital areas, and now these herpes viruses are spreading all over the body. Same thing with shingles. Usually it's a rash on the chest or back, and now you're looking at that virus spreading to the extremities and causing severe disease. Various types of rare cancers. You can see here's one here called Kaposi sarcoma. This is a cancer of the blood vessels. Normally, people's immune systems are keeping this in check, and you're not getting this kind of disorder. A few other diseases. Pneumocystis. So what is that? That is a type of pneumonia caused by fungal infections. Usually healthy people don't get these. Tuberculosis. This is not so common in Canada, but people with AIDS in Africa, often tuberculosis is a common secondary infection. Thrush as well, usually found in infants, but if you have AIDS, then it may actually be knocking down your immune system and allowing you to get thrush. So let's go back to the HIV virus. Here it is. We talked about it before, way back in topic 6 when we were talking about viruses. So remember, the HIV virus has these things here. These things, GP120 and GP41. These are glycoproteins, so glycoproteins. And GP120 right here, this is a docking protein, so it actually attaches to receptors on T helper immune cells. So these T helper immune cells are immune cells that actually activate other immune cells as a part of the adaptive immune response. And they have receptors, and these receptors are called CD4 receptors right here. So notice other aspects about the HIV virus. Remember, we talked about this reverse transcriptase. We'll come back to that in a minute. That's important for therapy against HIV. So here's the HIV life cycle. I am not going to review it in any detail here. You can go back to your topic 6 notes, or you can check out this video here. I will also add the video to the course playlist for this lecture so that you can review it and take a look at it. But I want to point out a couple of things here. Here's the HIV virus, and notice right here we have these CD4 receptors. Let me circle them a little bit better. There we go. These CD4 receptors right here are being recognized by the HIV virus. Also notice here we have the reverse transcriptase. That is converting the viral RNA right here into DNA. And then the last part over here in the nucleus, remember, it integrates into the host genome, and that's the first part of the life cycle. Eventually, the virus RNA gets expressed. You can see it's leaving here through the nuclear pore, gets converted to proteins. Those proteins assemble into new virus particles. You can see over here on the left, the mature virion has been budded from the cell to infect the next person. So check out the video if you need a refresher. So let's take a look at the epidemiology of HIV transmission. So the virus itself is infecting these CD4 cells. These are immune cells, of course. They're found in the blood and the lymph. So if you take a look at this list, it's found in the spinal fluid, the intestinal mucosal secretions, and the most important bodily fluids are the blood plasma and semen, in that the virus is found in significant amounts. Also found in ear secretions, but not usually a risk for infecting other people. So what are the consequences of this? This means that the spread of HIV is through direct contact with these particular bodily fluids, in particular the plasma, the blood plasma, and semen. So what bodily fluids are important? Blood and preseminal fluid and seminal fluid are the most important fluids to be concerned with HIV transmission. We're also concerned a little bit about vaginal fluid and breast milk. You can see small numbers, vaginal fluid and breast milk here, less than 1 particle per milliliter, but it is enough to be concerned for transmission from one individual to another. So what does this mean for transmission? Transmission includes, of course, sexual intercourse. We're talking about oral, anal, and vaginal sexual intercourse. Blood transfusions, hypodermic needles, so drug use, prenatal, childbirth, breastfeeding, all of those are things that are at risk. Somewhat in this order, sexual intercourse, blood transfusions, and hypodermic needles are the highest risk groups. We are, of course, concerned about transmitting to our children as well. And so there is some risk for people with HIV. Considering breastfeeding, for example, is done daily, even though the risk is low, the daily usage may actually cause a little bit of risk. So what else can we say about the epidemiology of HIV transmission? So take a look at this. This is the different types of transmission between males and females. And if you take a look at the differences here, this is at least for Canada. In males, a huge proportion of HIV transmission, 67%, about 2/3, is male-to-male sexual contact. The other big parts, of course, 15% heterosexual contact and 13% injection drug use. And then you can see that other uses are the minority. In females, over 80% of the transmission is heterosexual contact and 19% is in injection drug use. So you can see the big risks for HIV transmission is sex and drugs. There's a lot that could be said about this from a sociological perspective. The main thing I want to say about sex and drugs is that these are unfortunately 2 areas in life where people do not always think straight. In practice, people can talk about safe sexual practices or not sharing injection drug needles, but then people don't do it. And like I said, with sex and drugs, people are not necessarily always thinking straight. And so this makes a huge challenge in trying to combat this disease in particular. So what else can we say about HIV transmission? If you take a look at this chart, this is looking at the amount of viral RNA. So you can see over here on the left axis, HIV RNA in the blood plasma during time. So this is weeks and years after transmission. So if you take a look at this particular graph, you can see in the first few weeks of transmission, so about 8 to 10 weeks, so right here, we have this part of the curve. The viral load in the blood is actually relatively high. So what is the consequence here? The consequence here is that this is the part of the cycle of the virus where people are at the highest risk of transmitting to other people. That is, within a few weeks after being infected. You can see eventually the immune system does mount an immune response. So these other parts of the life cycle, you can see actually for several years the immune system is actually keeping the viral load relatively low in the blood. Although of course with sexual contact people are doing this repeatedly and they may not get HIV from having sex once or twice or even 10 times with an infected person. But if this continues for years, the chance of getting infected does increase. It can happen the first time. Again, it kind of depends when and how recently the person has been infected, what the actual risk is. Eventually, you can see at about 8 to 9 years, the viral load starts to increase. This is where the immune system can no longer keep the virus at check, and the person is going to start to get actual clinical AIDS. And we'll talk about that in a minute. So this is the key message. HIV transmission is most likely to occur with someone who is newly infected. Like I said, within that about 8 to 10 period after the person has been infected. So let's talk a little bit more about HIV transmission. So if you take a look at this pie graph on the left, I know it says USA, but of course this is referring to most Western countries such as Canada and European countries, in that HIV transmission is from in males, primarily the big chunk is homosexual male contact, in females heterosexual contact, and of course intravenous drug usage. So one question I have for you, how does this information affect prevention strategies? So you probably know a little bit about this, in that there's lots of education, particularly for HIV, that is targeted towards people who are involved in homosexual male relationships, and of course people that are drug users and sex workers and things like that, and of course people who may be new to sexual contact, such as high school students or college students. And so there's a lot of education on safe sexual practices and things like that. The interesting thing, if you take a look at the second pie graph, so this is looking at the world, so you can see that much of the world, heterosexual contact is the primary means of HIV transmission. So this is mostly referring to developing countries where heterosexual contact is the main way that HIV is spread amongst individuals. So this can have an effect on different programs that are going on in different countries. There's a lot that could be said about this. It's really very, very complex if you take a look at the issues going on in different countries. Some countries like Thailand have a lot of sex workers, and so that turns out to be a big part of the HIV transmission there. Places like South Africa have a lot of migratory workers that may have families at home, but they don't see them for 3 or 4 years, and so they're making use of sex workers. South Africa, unfortunately, as well, has a very high percentage of rape, which can contribute to transmission of sexually transmitted diseases. So here's a map of the world. I know it says 2011. Not much has changed since then. Maybe the numbers have changed a little bit. You can see Canada is in a very good place to be, where a very small percentage of our population is infected by HIV. You can see that different parts of Africa tend to have much higher numbers. South Africa, I believe, has the highest number of HIV-infected individuals on the planet. Again, much could be said about this. I'm not going to go into it in a lot of detail, trying to keep the lecture at a manageable time. I did want to show you this model for HIV transmission that actually shows some of the differences that happen amongst different populations. So if you take a look at the top box here, I have something called serial monogamy. So this is kind of generally what happens in Canada with relationships, sexual relationships. So if you take a look at this, we have a bunch of couples there. So these couples could be heterosexual or they could be homosexual, doesn't really matter. But you can see that, for example, take a look here. I have one person that has become HIV positive, and he or she may spread it to his or her partner. So there it goes. And that's usually the end of the transmission cycle in many cases in Canada. People are together, they're together sexually, and usually they're only together sexually with one other individual at a time. And we have good health care. We find out we're HIV positive. We get treatment, and we use that information for when we have new partners. So these people may break up eventually, and then they may end up in another monogamous relationship. Take a look at the chart down below. We have concurrency. So concurrency can happen in a number of situations. So for example, you could have a sex worker that is seeing many different partners or clients, or however you want to phrase it. And if the sex worker, so here we'll pull one up here, becomes HIV positive, or at least in this case it looks like somebody's partner is becoming HIV positive and then spreading it to his or her partner. And that partner, in this case, looks like has another sexual partner, which is spread, and it's just really this big web where many people can get infected simultaneously. So like I said, this can happen amongst sex workers or different cultural areas where, for example, there's a place in Kenya where polygamy is practiced, and so people are sharing wives and things like that, which can lead to, of course, many infections very, very quickly. you want to read more about this, check out this book. This book here is very, very good book. Very interesting. I believe we have it in the Canada Library. I encourage you to check it out. Talks a lot about HIV and some of the things going on in different parts of the world. So let's talk a little bit about the risks to healthcare workers. Of course, healthcare workers will be sometimes exposed to patients that have HIV, and so of course you're going to take preventative measures. The usual preventative measures, of course, are personal protective equipment, gloves and goggles and things like that. Anytime that blood is going to be an issue, such as surgery or something like that, extra precautions are taken. Of course, dealing with equipment and sharps and those kind of things, extra careful handling, and making sure any time that blood is an issue, it's cleaned up immediately. What happens upon accidental exposure? So typically, a person is put on antiretrovirals immediately in high concentrations to try to eliminate any virus that may get a chance to infect the individual. It does happen. The risk is relatively low, but it does happen on occasion that healthcare workers do get infections from their clients, and it's unfortunate, but it is a small risk being on the front lines. So let's take a look back at this curve again. I want to talk a little about progression of HIV infection. So back to this curve here. This curve is showing the viral load. So this is actually detection, you can see here, of the viral RNA. That's one way to detect the presence of the virus in the blood. So you can see initially a person gets a big peak in the virus particles, the immune system is keeping it at bay for a number of years, and then eventually the virus starts replicating again in high numbers. So let's take a look at what's going on in the immune system. So if you take a look at this, we have a few different types of immune cells that are being tracked. In this first part of HIV infection, so notice I'm looking right here, these CD4 cells are taking a hit. So they're being killed by the virus, and so at this stage in the viral disease, people usually end up with kind of like a mono-like infection. So then with fever or chills or something like that, extreme fatigue, maybe a little bit of weight loss, it goes away after a few weeks. This is why some people don't know they're infected. They think it's mono or just some sort of other illness. You can see we're also following these CD8 T cells. These are another type of T cell. And that initial immune response, that's where you get the fever and all that, and then they kind of plateau. So if you take a look at this chart, these CD4 cells are slowly being killed off. It takes a few years, but the HIV virus is slowly killing them, and some people it's a little bit shorter, some people it's a little bit longer, but typically after about 8 years of slowly having declining immune response, which means that people are going to start to get a little bit more infections, maybe a little bit more serious from things like the flu or pneumonia or things like that. Usually not life-threatening at this stage, but their immune response is decreasing. At a certain point, so we're looking at right here. So at a certain point, their CD4 cells decrease to an amount that their immune response is quite crippled. And this is where what we have, we call this clinical AIDS. So clinical AIDS means that that CD4 count is at a certain level and they're starting to get the presence of certain rare infections. So we talked about those rare infections already. We talked about disseminated shingles, we talked about thrush, tuberculosis, and there's a whole list of various diseases that people will get when they hit AIDS, clinical AIDS. Here's a note here. It says, in the absence of antiretroviral therapy, life expectancy is about 10 years. And the median survival time is after developing clinical AIDS is 9.2 months. So here is the AIDS diagnosis. So this is a clinical diagnosis of AIDS. So someone who is HIV positive does not have AIDS. They have AIDS when they have this. So notice all these things together. The presence of rare diseases. So there's kind of a list of rare diseases. Something that used to be on that list was actually toxoplasma. Nowadays we know what to look for it, and the person's often treated for it before they even get to the AIDS diagnosis. Unexplained weight loss, fatigue, and fever. But here's the other key thing. Less than 200 CD4 cells per microliter in the blood. So these two things in particular, the CD4 cells, the rare diseases, and I guess the other thing that's not on this slide, I think it was on the previous slide, is the presence of the HIV virus. So don't forget that one as well. That is an important diagnosis of somebody having AIDS. Here's some of those rare diseases. Huge list here. Oh, there's toxoplasma on that list. I just mentioned toxoplasma right here. Notice a bunch of others, cryptosporidium, leishmania, microsporidium. A bunch of these are things we haven't talked about. And the reason why we haven't talked about a bunch of these is because they're so rare. Most people are not getting them unless they have a huge immune failure, such as AIDS. Here's tuberculosis here. Notice there's other rare mycobacteriums that people can get that normally healthy individuals are not affected by. We got all sorts of weird fungal infections here, and of course various viral infections that can cause people problems. So serological diagnosis. So notice here HIV infection. This can be diagnosed by a variety of serological methods. So we have ELISAs, we have agglutinations, we have something else called a Western blot. So the ELISA is usually the first test. Nowadays we have those rapid tests, as I mentioned before, that can be done within minutes, or people can do tests that take 24 hours or so, depending on what's done by the clinician. Here's the rapid test. So as I mentioned, they're very similar to pregnancy tests. Notice you have a control band. This will show up on every test. If it doesn't show up in the test, it means the test is not functional. This person here is HIV positive, and this person here on the right is HIV negative. So what about treatment of HIV? Nowadays we're using something called HAART, H-A-A-R-T, highly active antiretroviral treatment. Highly active. This is named by the pharmaceutical industry. They like to, have buzzwords and fancy things to show how good they are at doing what they're doing. But basically, this is a cocktail. So these cocktails include things we talked about: protease inhibitors, nucleotide and nucleoside analogs, reverse transcriptase inhibitors. So you're probably wondering, why a cocktail? Well, there's one big issue with HIV. HIV mutates rapidly. This is a problem, very, very rapidly. You probably know that the flu virus mutates rapidly, about once a year. HIV does this within weeks or a month. And so a person can be have an immune response against one HIV particle, and it's useless a month later because the virus mutates so rapidly. So we use multiple drugs because we don't want the virus to become resistant to one drug. If it becomes resistant to one drug, it becomes useless. So we kind of hit the virus at different angles with different drugs to try to prevent any mutants from arising and becoming resistant. So you're also probably wondering, what about an HIV vaccine? So here's the virus again. Remember, we have these glycoproteins on the surface. These are the most likely vaccine targets for the immune system. Again, remember that problem. HIV mutates. This is a big problem. That reverse transcriptase is very error prone. That means it makes mistakes when it copies the RNA genome. And this leads to all sorts of rapid mutations. So this is why it's very hard to mount an immune response against HIV, never mind the fact that it actually attacks the immune system, which makes the response even more difficult to propagate. We do have an immune response. There's lots of really interesting research on this that I'm really not going to dwell on this particular semester. So I do want to share one other story with you. This is about a man who was initially known as, you can see on the slide, the Berlin patient. His name is Timothy Brown. He was an individual who had HIV and ended up somewhere along the way developing leukemia. So you may know that a treatment for leukemia is getting a bone marrow transplant, and so he was able to do this. He got a bone marrow transplant, and the very interesting thing about this particular patient is that his donor was one of those rare individuals who's actually resistant to HIV. So about 1 in 100 humans has a mutation in their CD4 receptor that makes them immune to HIV. They can't get it. And so he was lucky enough to get a transplant from this person, and of course a bone marrow transplant is basically an immune system transplant, because that's where the immune cells are made. So this was a very rare case. He was initially on antiretrovirals and went off of them, and they have not been able to find virus in his body for, I think this is almost 10 years now, which is just amazing. This is not an easy process to do. It's very expensive and very hard to find donors, never mind finding donors who are resistant to HIV. There's a second patient who came open in the last year. They called him the London patient. I think his name was finally released because he also wanted to share his story. But this does show that there is hope, and maybe someday there will be a potential cure. Maybe with some sort of genetic engineering or something like that, we will someday be able to do something about HIV and AIDS. So I just kind of want to finish off this topic and just say that, of course, AIDS and HIV is a very, very complex issue. As I mentioned, we're dealing with sex and drugs and all sorts of sociological issues. It affects different countries in different ways and affects different people in different ways. And it is something that I find very fascinating. And anytime you want to come talk to me about HIV and AIDS and pick my brain about what I know, please feel free to drop by my office. So that concludes topic 13 on the immune system. Don't forget to go through these concepts. Don't forget to go through the terminology and the study questions, and hopefully it's clear to you what you need to focus for on the final exam. Here are some text references. I want to point you to this. There's some additional references here online, and this is actually a pretty good reference if you're looking for more information on HIV life cycle and AIDS and related concepts. I hope you have a great day. We will see you in the next lecture.